Hi, today I will be presenting the COL1A1 gene to my mother who has no science background. So, hi Mom. So the collagen type 1 alpha chain gene, which is the long word for the acronym I just said, is very important in collagen production. So this mature protein can be found outside of the cell in this place called the extracellular matrix, but you can think of it as being manufactured in this organelle called the rough endoplasmic reticulum. And so this occurs in cells that are important in bone formation, connective tissue formation, such as osteoblast and fibroblast. So they create this strong extracellular matrix, which provides support for us. However, errors occur and it can lead to negative consequences. So we will discuss Caffi disease, which is a result of a mutation in the COL1A1 gene. Arginine is changed to cysteine, and it changes the structure of the protein, which then changes the function. And so in Caffi disease, we'll discuss more, but it is characterized by excess bone formation and inflammation seen in infants. So to show my protein, I made 2 models, or maybe 3. So to show the structure, this gene creates a triple helix molecule. So there are 2 alpha chains, which are purple, 2 alpha 1 chains, and then 1 alpha 2 chain. And they combine to make this triple helix. And this is known as a quaternary protein structure, which basically means different subunits of proteins come together to form a more complex unit. But additionally, these individual chains seen in here are just a primary protein structure. So it's just a single linear arrangement of amino acids in the most basic form of a protein. So in my model, I wanted to show the triple helix and arginine, which is our important amino acid. So with arginine at position 836, this amino acid is positively charged, hydrophilic or water-loving, and polar. And so it contains a functional group, sorry about that, a functional group called a guanidinium group, which allows these hydrogen bonds to form. Up to 6 can form, which provides a lot of structural integrity. And also, since this is positively charged, it can form salt bridges, another type of bond, with negatively charged amino acids and other molecules. So with this structure, we are able to get our uniform, strong collagen fibrils in places like our bones, skin, tendons, and ligaments. And so the white lines, like I said, represent the hydrogen bonds. Black line is our amino acid arginine, and you can see structurally sound. However, in Caffi's disease, instead of our arginine at position 836, we get a small amino acid called cysteine. It alters the structure of the triple helix because cysteine has much different properties than arginine. For example, rather than that guanidinium group that we saw, cysteine has this thing called a sulfhydryl group, which can form these things called disulfide dimers. There are still hydrogen bonds, but they're not enough to provide the support that we need. Cysteine is also hydrophobic, also known as water-fearing, and it has a neutral charge rather than positive charge we saw in arginine. And so with this change leads to irregularly shaped collagen fibrils due to these disulfide dimers. And so they're not structurally sound. We have an abnormal cell matrix, and this leads to the bone formation, swelling, and skeletal abnormalities seen in these infants with Caffi disease. So to demonstrate this mutation and issues with structural integrity, I tried to bend the protein, and also you can see there's interactions with these other molecules which further interfere with the structure. So obviously there are some limitations with my model. First, collagen fibrils and alpha chains are super small. And so this model is a very big dramatization of what those actually look like. And also, it doesn't show the complex chemical bonds that occur in our body. And finally, although I mentioned the hydrophobic and hydrophilic properties, they weren't able to be shown well.